Good morning. I am Dr. Mrs. Manju Sahai, a contractual faculty working in the Department of Botany, Mahila College, Chaiwasa. Today I deliver my lectures for semester 5, DAC 1. The topic is peroxisome and glyoxisome structure and function in animals and plant biogenesis. Now, these are not met in general cell structure. It is only present whenever its function is to be needed. These two are microbodies. When it is microbodies means glyoxisome is another one of the microbody and peroxisome is another microbody. Glyoxisome performs glycolate pathway, peroxisomes performs glycolate pathway. Glycolate and glycolate is aldehydic form of 2 carbon unit CHO, CH2OH, and the glycolate is COOH and CH2OH. Both are oxidized and unoxidized, matlab removal of oxygen means aldehyde. Dic group is glycolate, acidic group is glycosylate. Both these two microbodies has single layered membrane. It is not double layered as mitochondria, as endoplasmic reticulum, as chloroplast, Golgi apparatus has. But it has only single membranous structures. All the glycosome performs glycosylate cycle and peroxisome performs glycolate cycle. For glycolate cycles, there is the need of actually these two microbodies responsible for conversion of fat into carbohydrate. Then when we know that carbohydrates are, that is the hydrates of carbon, saccharide unit CN, and polymer of which is polysaccharide, starch storage in green plants, and glycogen in animal and bacteria and fungi. Now, ये carbohydrates is the easy source of energy. Carbohydrates, CnH2nOn itself is carbohydrates, monosaccharide itself is carbohydrate, starch itself is carbohydrate. But when fats converted into carbohydrates, fats are not being oxidized easily. Rather, it has to hydrolyze into fatty acids and glycerol. One molecule of glycerol come in contact with 3 molecules of fatty acids. Fatty acids are nothing but all monocarboxylic acids are called fatty acids. Fatty acids is a long chain carbon having the formula CnH2nO2. Means COOH is the monocarboxylic group at the first carbon, and next is the alpha carbon, next is the beta carbon, and so on. The only long chain even carbon containing saturated fatty acids are essential for further breakdown. Means for further breakdown to lead the formation of carbohydrates. In the first step, all the enzymes responsible for glycolate pathway are present in the stroma of glyoxisome, and also the glycolate pathway enzymes are located in the stroma of peroxisomes. It is single membranous, no doubt. It is not self-reproducing just like mitochondria or but their function is not singly alone exhibited. It is rather connected with mitochondria, connected with cytoplasm, connected with chloroplast. There are certain organisms may involved in the right working of this pathway. But first of glycolate pathway, glyoxisome में जो होता है fats, first hydrolysis comes fat, 1 molecule of glycerol and another 3 molecules of fatty acids of even carbon long chain. They go further oxidation that takes place in mitochondria. Fatty acid is inactive form. It has to become activated by the expenditure of energy ATP, coenzyme A, and its magnesium, they are taking in account for the activation of fatty acid. This fatty acid becomes active, means fatty acyl coenzyme A is now going to outer membrane of the mitochondria, then go to the inner membrane of the mitochondria. In matrix, they perform the beta oxidation of the fatty acids. Beta means carboxylic group, next is alpha, next is beta. That very beta carbon is oxidized. Now, in the first step, when fatty acyl coenzyme go inside the mitochondrial matrix, it shows further dehydrogenation in term of oxidation. When certain hydrogen acceptor bodies accepts hydrogen, this is oxidized and becomes enoyl coenzyme A. The FAD is a nitrogenous hydrogen acceptors. FAD means paradoxine adenine dinucleotide becomes reduced, FADH2, that going to respiratory chain in mitochondrial cristae, forming 2 ATP. Now this enoyl coenzyme A then hydrated by hydrolase and forming beta hydroxy enoyl coenzyme A. Then again another oxidation process occurs in terms of dehydrogenation. NAD is involved in capturing the hydrogen from the hydroxy enoyl coenzyme A. This NAD is used. NAD, the full form is nicotinamide adenine dinucleotide in oxidized form. It catches the hydrogen, becomes NADHH plus, and this on electron transport chain releases 3 moles of ATP. Now this 3-keto enoyl coenzyme A, then again there is thiolase reactions, expenditure of 1 molecule of coenzyme A, the cleavage of this into 3-hydroxy enoyl coenzyme A into 2. One is 2 less carbon atom, and the 2 carbon atom is liberated in active acetate form, that is the acetyl coenzyme A. So this is all about the beta oxidation. The further the fatty acyl coenzyme A residue, they again circled by beta oxidation. Every time it releases C2 unit, that is beta acetyl coenzyme A. If the fatty acid is palmitic acid C16H32O2, then it's by 7 times it becomes 8 acetyl coenzyme A. That acetyl coenzyme A is, when they combines with oxaloacetate, it forms citrate. Citrate is to forms isocitrate. Isocitrate goes to the citric acid cycle performs like this. But in case of Glyoxisome, this isocitrate lyase is cleaved, मतलब वो उसका lyase हो जाता है, टूट जाता है, that is in the form of C2 unit, that is glyoxylate and the succinate. Succinate goes to the mitochondria for further oxidation, means succinate, fumarate, malate, oxaloacetate, then again acetyl coenzyme forming citrate, isocitrate. This is the Krebs cycle. Now this glycosylate then combines with another molecule of acetyl coenzyme A, the malate synthetase. Formation of malate is going on, and the liberation of coenzyme A. This is the 2 enzymes. One is isocitrate lyase and another is malate synthetase, the key enzyme of glycosylate pathway, which a glycosome actually in germinating seeds, they are in function because seeds are often rich in fats. Fats have to form glucose, means fats, easy way of energy liberation is the carbohydrate substrate. So fats conversion must be there into glucose, and it is via glycosylate, mitochondria, and then the malate formed inside the Krebs cycle, inside the mitochondria, released, diffused to the cytoplasm, where it is decarboxylated means phosphoenol pyruvate carboxylase CO2 liberation forming phosphoenol pyruvate that going to reversal of glycolysis that is known as gluconeogenesis forming glucose by fructose 1,6-diphosphate. Fructose diphosphate 1-phosphate is going to convert ADP to ATP, and the fructose 6-phosphate is now formed. Fructose 6-phosphate is combines isomer is of which is glucose 6-phosphate, and then the two combines forming sucrose. Sucrose is the non-reducing sugar. All monosaccharides are reducing sugar. Reducing sugar can't move from one place to another. Non-reducing sugar move from one place to another where there is the need of energy. Sucrose move, and when energy is to be taken, sucrose is degraded into by invertebrates from glucose and fructose. Glucose by normal glycolysis forming pyruvic acid. The C2 unit forms decarboxylation in form of dehydrogenation oxidative, and the C2 unit comes in mitochondrial releases ATP. And in Krebs cycle we know 12 ATP per acetyl coenzyme is liberated. So huge amount of energy is liberated by the oxidation of palmitic energy. That very energy is utilized for the growth of seedling at the time of germination, because seedling has to perform a huge plant, tree or shrub or herb, may be the case, but it has to form a new plant from single zygote embryo. These are needed. Thanking you for details. Kindly consult in my PDF.